In this part of the lecture, I just want to briefly touch on some of the practical considerations for when you're planning your own single-cell RNA-seq studies. And perhaps the most important thing to just keep in mind and ask yourself is really whether you even need to do single-cell RNA-seq. In other words, do you need single-cell resolution to answer the question that you're setting out to address? And again, the question should drive the use of the technology, not the other way around. So you should think a bit about whether something like flow cytometry or bulk RNA-seq would actually answer your question, or at least get you sufficiently towards an answer to your question. If so, these are techniques that, as you're going to see in just a minute, are far less expensive than single-cell RNA-seq, and they involve oftentimes, but not always, a lot less downstream bioinformatics analysis. So when is single-cell RNA-seq preferred? Well, the first thing to keep in mind is single-cell RNA-seq is not a replacement for bulk cell. It's not better than or greater than, but it's different. It allows us to ask different kinds of questions. And so if you have a question that really hits at heterogeneity in a tissue, for example, then absolutely single-cell will be a much better tool for addressing questions related to heterogeneity than bulk cell would be. In a similar way, if you care a lot about the developmental trajectory of cells within a tissue, how one cell type gives rise to another, single cell is excellent for that because we can detect all these subtle differences in transcriptional states of cells, and we can even correspond one state to another through methods we don't have time to get into in these lectures, but suffice it to say, single cell is great for that and bulk cell is not. If you care about multiplexing, as we saw in the last part of the lecture, single cell lets you pair multiple different assays all at a single cell level, whereas we really can't do that at a single cell level with bulk. And so this might lead you to believe that single cell is greater than bulk, but there are some important advantages that bulk has. And probably the first and foremost is that bulk RNA-seq gives us a far deeper profiling of the transcriptome in a tissue. We'll get data for perhaps all the genes or transcripts in the genome, whereas we may only get 1,000 or 2,000 or a small subset of the transcripts or genes profiled in a single cell experiment. And that comes at a far greater cost, actually almost an order of magnitude more expensive per sample to profile at the single cell level versus to profile at the bulk tissue level. And then, as I already mentioned, the analytic burden for single cell is generally speaking much, much higher than it is for bulk cell, and that is even more so the case if we're talking about multiplexed experiments. So save yourself the time. Don't be Michael Scott. Don't make this way harder than it needs to be. All right, in the pre-course lecture, I talked just briefly about the costs of bulk RNA-seq, and I even had this asterisk here that costs associated with single-cell RNA-seq were just much more complicated. And that still is the case, but I want to try to disentangle that just a little bit and dig into this asterisk. And so if we talk about costs for single-cell RNA sequencing, we're still talking about library prep reagents and the actual consumables for sequencing. And I'll sort of paint for you below sort of best case scenario for costs so that you understand how this compares to bulk RNA-seq. Again, I'm talking about the 10x Genomics platform, and I'm going to be talking about sequencing on a NextSeq 2000, which offers a very sort of appealing and affordable way and flexible way to sequence single cell libraries because we have two main outputs. We can output 400 million reads, or we can use the P3 flow cell to output 1.1 billion reads. So this lets us profile a small number of samples by single cell or a larger number of samples. So the complications in estimating costs for single cell RNA sequencing is that it depends on how many cells we encapsulate, it depends on how deeply we sequence each cell, and it depends exactly on the kind of library we're making and on the kinds of consumables we use on the sequencing end. So there are a lot of assumptions baked into this slide, but at least it's a point at which you can start to wrap your head around costs. So if we're talking about 10x Genomics, those kits for library prep come in 4 reaction and 16 reaction options. The bottom line is at a per sample cost to encapsulate a sample in droplets and then prepare libraries from those samples will cost you roughly in the neighborhood of $2,000 per sample. So that's in stark contrast to bulk RNA-seq, which might be 1/10 of that cost. There are newer options from 10x, the so-called low throughput or LT kits, that let you profile, instead of 10,000 samples as we would do with the kit here at the top, let you profile 100 to 1,000 cells at a smaller, although still not trivial cost. Once you sequence, depending on whether you're sequencing on a P2 or a P3 flow cell, remember, we have to do paired-in sequencing. We can't get by with 75 base pairs, so we're going to have to pay for more sequencing. One nice thing about the NextSeq 2000 is that we can get by with just 100 base pairs because each of their kits actually has reagents sufficient for an additional 36 to 38 cycles. I can't remember exactly how many. And so that helps us keep our costs down. We don't have to use a true 150 cycle kit. We can just use their 100 cycle with these sort of bonus reagents, if you will. And so those flow cells come at a fixed cost of $1,400 or approximately $3,200. Again, these are ballpark figures. Bottom line is, if you want to profile 2 samples at 10,000 cells per sample sequenced on this P2 flow cell, you'd be looking at about 20,000 reads per cell at a cost of close to $2,500 to $3,000 per sample. Now that cost would come down a bit if you used a P3 flow cell and piled on 5 to 6 or perhaps more samples. Your sequencing cost would come down, but you're still going to be north of $2,000 per sample to carry out single-cell RNA-seq. All right, so what are some considerations for a successful RNA-seq experiment? Let's say you can afford it. You've decided that it fits or is the best tool to address the question you have. What should you consider going into that experiment? If I could give you one piece of advice, it's to think carefully about optimizing recovery of cells to maximize cell viability. And I can't emphasize this enough. Healthy, happy cells are critical to the success of single-cell RNA-seq. Ideally, viability should be above 90%. If it's not, you're already tilting the scales against a good experiment for single-cell sequencing. Why is cell viability so critical? To illustrate that, I'll use this wonderful video from the Microbial Ecology Instagram account that shows a protozoan cell dying, just to illustrate that when cells die, they spill out their contents everywhere. We can see stuff spilling out here, stuff spilling out down here, and up here. Okay? And when that happens in the context of a single-cell RNA-seq experiment, when this cell is dying in solution before encapsulation, all of these extra bits that spilled out end up distributed throughout your aqueous solution and get into every droplet you make, even droplets, and this is critical, that have no cell in them. And so they become something we call ambient RNA. RNA that's in your environment, in the buffer, in the solution, that becomes background and makes it very, very difficult to tell your droplets with cells apart from droplets without cells. And just to illustrate how frustrating ambient RNA can be, it doesn't take an extravagantly unhealthy, dying cell population to really complicate things. You can have a single cell type in your tissue that is particularly prone to cell death, and that can have effects on everything else you see in that experiment. So as an example of this, and this is all courtesy of Owen Whalen's work in Ralph Brinster's lab at Penn Vet, I just want to highlight one of the experiments that they've done where they can take mice that are unable to generate spermatids, so mature cells in germline differentiation in the testes. They know that these mice can't generate those cells, but they can inject those testes with GFP marked cells that restore the full competency of germ cell production here. And when they do that, the testis gets larger, and you get full spermatid formation. And so when they do single-cell RNA-seq on those testes, you can actually look at the control that hasn't received the transplant, and if you look for a marker of spermatids, you see no expression in any of your clusters of cells. However, and this is where the ambient RNA becomes a critical aspect, is when you look at the single cell data from the transplanted tissue and you ask where protamine 1 is expressed, it should only be expressed in spermatids. And indeed, we see a large population of spermatids here that were not in the control samples. That's great. But the concerning thing is we now see expression of protamine 1 leaking into all of these other cells, and we know that that's not real expression. It's not present in the cells from tissues that did not receive the transplant. Why? Well, one explanation for this is that these more mature germ cells, these spermatids, are more prone to cell death. Their RNA leaks out and becomes ambient RNA that you pick up in every droplet, regardless of the cell type that's in that droplet. Sperm and germ cell populations are not the only problematic cell type. A recent paper, actually the one describing T-seq that I talked about earlier, highlighted that neutrophils, a cell type in the immune system that we know is quite adept at rapidly dying and does so as a way to control infections and inflammation, that neutrophils can really complicate clustering and identification of clusters from chromatin accessibility data. So here you see unsorted cells where there's been no removal of dead or dying cells. Removing dead or dying cells may help your clustering a bit, but removing neutrophils dramatically improves clustering. And the tightness of the clusters is much greater, and you even have identified aspects of the clusters like activated B cells or like different monocyte populations that were simply not visible prior to removing neutrophils. So this is all just to say that depending on the exact tissue and the context of your experiments, there may be cell types that you want to think carefully about and strategies that you may employ to help get the best quality single-cell RNA-seq data or single-cell genomic data that you can get. All right, so I'm going to summarize some of the recommendations that we've learned for approaching how to do a successful single-cell experiment. Again, we're learning too, and so our recommendations will change certainly as we learn more. But right now, I think I can say that cell isolation protocols that may have been optimized in a lab for flow cytometry or even in vitro functional assays may be insufficient for single-cell RNA-seq experiments. And that's because we could have a fair number of dead or dying cells and still get good surface staining or intracellular staining by flow cytometry. We could actually still get enough live functional cells to get a good readout in our in vitro functional assay, but these cells could be a disaster by single-cell RNA-seq if they're leaking RNA out into solution. If you're accustomed to digesting tissues at 37 degrees Celsius, which we all have done, you should consider switching to a 30-minute digestion at 6 degrees Celsius with something called a cold activated protease, and I've linked to the paper here. If you need a starting point for tissue dissociation protocols, if you're embarking on a new experiment or have never worked with a particular tissue before, the Worthington guide is a good place to start for guidelines on that. And then at all costs, avoid mechanical dissociation. Don't bash up your lymph nodes or spleen with a syringe plunger. Use less harsh, less mechanical methods for separating tissues. Regardless of how you get your cells, whether you're doing bead-based purification or flow sorting, you really should carry out a dead cell removal. And I've linked to a kit here, and really any kit for this is probably sufficient, but the goal here should be to get viability above 90%. Bead-based purification of cells is preferred over flow sorting in most cases because it's usually faster and it usually results in happier cells. Depending on your experimental goals, it may be totally fine if purity isn't 100%. Remember, you're going to get separation of cell populations by single-cell seq, so it's okay if you have a contaminating cell type in many cases. I would prefer that over extended periods of time flow sorting an exceedingly rare population that ends up being unhealthy. Keep everything cold, cold, cold. Cells should always be kept on ice, even when walking from the bench to the centrifuge. Buffers should be pre-chilled. Centrifuges should be pre-chilled. This will all help limit cell death and clumping. And clumping is also really bad because it means a potentially clogged microfluidic device. And then finally, you may want to avoid using EDTA if possible in your tissue dissociation buffers. Excessive amounts of EDTA greater than 0.1 millimolar or magnesium greater than 3 millimolar will inhibit reverse transcription reactions and can lead to reduced complexity in your single-cell RNA-seq data set. Okay, as I said, we're still learning about single-cell protocols. Any protocols that we have are available on our protocols site, which I linked to in the slides. We have a whole section on single-cell genomics, and you can always check out both our frequently asked questions if you're curious about the technology in general, and much of this was just covered in the lecture, or specific protocols like site-seq or 10x. Okay, so heading back to the presentation. And then another major consideration is what you'll do after you get data, the data analysis phase of things. And so I want to just point out that the main approach most people use when they've generated data on a 10x platform is to use the software developed by 10x called CellRanger. And CellRanger takes the raw sequence data and identifies barcodes and UMIs and pre-processes your data so that you get a table of cells as columns and genes as rows. But CellRanger is really resource intensive. First of all, it only runs on a Linux system. It requires at least an 8-core machine, but 16 cores is recommended. You need at least 64 gigs of RAM, but again, 128 gigs of RAM is recommended. This is a lot of RAM. You need at least a terabyte of free disk space, and I would argue actually far more. And so these all mean that this really is quite a burden and one that you're not going to be able to address on a laptop. So what we'll use in the next part of the lecture is a recently described set of tools based on Callisto that allow us to process the same raw data, but to do so on our laptop. And they highlight in this paper, this is Paul Melsted's work when he was in Leora Pachter's lab, that another challenge in single-cell RNA-seq preprocessing is the amount of data that must be processed. A typical single-cell experiment can generate 10 to the 6th to 10 to the 10th reads from 10 to the 3rd to 10 to the 6th cells. This is leading to bottlenecks in analysis. For example, the current standard program for preprocessing 10x genomics chromium single-cell seq data, the CellRanger software, which I described here, requires approximately 22 hours to process 784 million reads and uses 1.5 terabytes of disk space. Not going to happen on our laptops, folks. So we're not going to use CellRanger. Instead, we'll use the work described in this Nature Biotech paper. And that work really is an extension of Callisto. You're already familiar with Callisto from lecture 2, the idea of pseudo-alignments and finding equivalence classes or sets of transcripts that are compatible with the read. That same sort of rationale underlies the Callisto bus tools approach, in which the process of read mapping is carried out by Callisto and is really inefficient, is really independent of the technology platform. And then the data that results from that read mapping is represented in a simple, relatively simple structure conceptually of barcode UMI set file, where you have a barcode represented as 32 base or less than 32 base barcode, and UMI represented using 2-bit encoding, and then 32-bit encoding used to identify the equivalence class and the counts, and then additional flags if needed. And we won't have time to really go into great detail about this bus format, but suffice it to say, it lets us use a read mapper we're already familiar with, Callisto. It lets us move away from CellRanger and actually carry out this whole process of pre-processing all the way through analysis in R on our laptop. And so it's great for us. One thing I'll highlight about Callisto bus tools is, as you might expect, because it's working with Callisto and because it uses this relatively simple and platform independent way of representing the data as a bus file, it's really fast. And so if we look at processing time in seconds, Callisto is the orange dots here, and we see that it is approximately 50 times faster than Cell Ranger, which is represented in blue. And it maintains a constant memory need. So you can see that the orange dots pretty much hover around 5 or less gigs of RAM used. As the number of reads increase, it stays in that general range of 2 to 10 gigs of reads of RAM used, and that's about the amount of RAM we can expect to use on a laptop and not have major issues. A lot of these other methods are also relatively fixed memory requirement, including Cell Ranger, but have far greater memory requirements. Okay, so we're going to now transition away from lecture slides and actually get down to the business of using Callisto bus tools to pre-process raw single-cell RNA-seq data directly on our laptop.